You have to note down only 2 or 3 points, not much more than that about these branch chain amino acids. And one thing I will tell you about newborn screening, that's something I have told you earlier. Tomorrow it will be a slight bit repetition of what I said earlier. So, can you hear? Can you hear properly? Okay. So, can you remember what are the branch chain amino acids? Anybody? What are the branch chain amino acids? Amino acid classification I told you. Start telling, at least start interacting so that your first internal assessment examination will be held in the next month on, I think, on 10th. So you must be prepared. Start revising whatever you have been told, otherwise you will not be able to cover. So branch chain amino acid name. Anyone? So you have not opened the book at all. I mean, it will be difficult. So why branch chain? Because there is branch, some amount of branching is there. The basic amino acid structure RCH, NH2, COH. On top of which only one methyl group is added, that is not included. If some more amino acids are added, then it is called a branch chain amino acid. So chemistry I am not going to tell because I have told you in detail, only thing is you have not read. Metabolic fat, I will tell. Metabolic fat is basically will involve catabolic part, not the anabolic part. Because branch chain amino acids are all essential amino acids. So they need to be supplied in the diet, so there will be no endogenous synthesis of the essential amino acids. So I will deal with the catabolic aspects only, and the steps of branch chain catabolism are many, but I will concentrate only on 3. That 3 steps you must remember, out of which one step is most important and one short note is likely to come, and that I will discuss in detail. That you note down almost all the points so that you can write the entire things, at least one question completely, because everything is not available in your book. And the disorders of branch chain background, that is the actually question, that one question is likely and this will be asked. So once you know the steps of catabolism, then you will understand what is the problem and why the disorder takes place, at least particularly one. And one name of the enzyme you have to remember, that is today's class. So branch chain amino acids are 3: Valine, Leucine and Isoleucine. You see here I told you the basic structure of amino acid is like this. This is from the chemistry class. Whatever possible things were there, I have told you in detail in the chemistry class and the previous classes also. So that if you remember all those things, it will be easier for you to understand. So only thing is that you see here, this structure is common. Only if CH is there, that is not included in that branch chain. Only 2 more carbon groups are there. That's why it is called a branch chain amino acid. And you see here, one more structure, a carbon group is there, the methyl group is there. And the isolation term is that because there is a simple position change, nothing more than that. The structure remaining same. That's why it is called isoleucine. It is an isomer of leucine. So all these 3 are essential amino acids. Okay. So, during amino acid class, every time I told you whether it is a glucogenic amino acid or whether it is a ketogenic amino acid. The basic purpose is that during your neoglucogenesis class, I think this has been covered, and one of the important aspects of gluconeogenic precursor will be the amino acids. So, those glucogenic amino acids which will enter into the gluconeogenic pathway will be basically glucogenic amino acids, and you have to remember I have told you what are glucogenic amino acids, what are ketogenic amino acid. At least one ketogenic amino acid you must remember, that is leucine, and lysine is also exclusively ketogenic amino acid in human beings. So, leucine you must remember, lysine is you also tell, and aromatic amino acids like phenylalanine, tyrosine, tryptophan, these three are glucogenic amino acid, on top of which isoleucine is added, and lysine also contributes to some extent to glucogenic amino acid. Rest all 14 amino acids are glucogenic amino acids. So, if I ask you, then you will not be able to tell, because since you don't remember the name of the amino acids, it will be difficult for you to remember. So, valine is basically glucogenic, leucine is exclusively ketogenic, and isoleucine is glucogenic and ketogenic also. So, why I tell every time this thing? Because you see, during the extensive, extreme phase of a body situation, like extreme, when there is a prolonged fasting, starvation, then the amino acid will be broken down, and these amino acid will serve as a fuel to the brain, particularly glucose and the ketone bodies, and that's why the protein will be broken down, and protein will enter into this, will be broken down into amino acids, and glucogenic amino acid will enter into the neoglucogenic pathway, and it will provide glucose, and ketone bodies will also add nutrition to the brain. That is the reason I have been repeatedly telling you. So they are alternate source of fuel to the brain, especially under conditions of extreme condition like starvation, or when there is a severe wasting. So, this trust every time I am telling, and this you try to remember, that is the basically metabolic fate of the carbon skeleton, and that involves all the amino acids, and in the gluconeogenic pathway, this will be a very good pathway to describe the gluconeogenic pathway, and then the center you see the TCA cycle. There are all 7 metabolites which will enter from different sources. Now, if you look at this thing, if you remember this chart, you see the leucine. Leucine is here. Leucine is basically producing acid to acetyl-CoA. Any amino acid which is producing acid to acetyl-CoA will give rise to ketone bodies, and acetyl-CoA can also form, but acetyl-CoA can form ketone bodies, but acetyl-CoA mainly is used in the fatty acid synthesis also. And isoleucine, you see here, it is producing acetyl-CoA, it is entering into the TCA cycle by this pathway, and also it is entering into the ketone body formation. Now, leucine is basically going to the acetyl-CoA. So, again leucine is basically and exclusively this leucine is here, leucine is here. So, these basically are entering into the acetyl-CoA, and this is exclusively ketogenic. So, you remember this part I have told you during your class, at least aromatic amino acids you remember. This is producing both glucogenic and ketogenic, that you must remember. Then catabolism, only I told you that many reactions I will show it to you, but only you remember these 3 steps. These 3 steps are common. First step is transamination. First step is transamination. The second stage is oxidative decarboxylation, and third stage is dehydrogenation. The first stage is transamination. So, enzyme concerned is called either a transaminase or an aminotransferase, as I explained you in detail during transamination reaction. So, either it is a transaminase or it is an aminotransferase. Both are synonymous. It is a freely reversible reaction. And don't forget to remember, this is a vitamin B6 dependent reaction, that is pyridoxal phosphate. This is called PLP. So, all transamination reactions are vitamin B6 dependent reaction, and see, if alpha amino acid is a transamination reaction involves one alpha amino acid and one alpha keto acid. Transamination reaction, this alpha amino acid gets converted into alpha keto acid, and this alpha keto acid gets converted into alpha amino acid, and these are reversible. When they move in one direction, you can say it is a transaminase. When this can move in the other direction, it can cause aminotransferase. It is the same. You have to see which substrate is there. That's it. Even if you write in this direction, if a reaction catabolizing from this direction to direction, aminotransferase name, there is no harm in it, and the vice versa. So, we will get at keto acid in the first stage. So, when we produce keto acid, so keto acids will be basically producing ketosis, ketoacidosis, and that will see if there is a block. If there is a subsequent block in this next stage, it will be, first stage is dehydrogenation, this is transamination, second stage is decarboxylation. Means carbon dioxide will be removed, and third stage, next stage will be basically dehydrogenation. Basic point of telling this thing is that if the reaction is not allowed in this way to produce its final metabolic product, if it is blocked somewhere here, then these acids and the main amino acid will be accumulated, and their levels will be high in the blood, and they will be filtered also in the urine. So, you can detect a high amount of this alpha keto acids and the corresponding amino acids. That means amino acid and their cognate keto acids will be produced in high amounts will be available in the body, because they are unable to be further broken down, because these are essential amino acids. So, we will take in the food. Do you get my point? Now I am moving further. So this enzyme, it's a basically one, write down this name. This is a multi-step process except transamination reaction. Except transamination reaction, for which there is a transamination, there is a mitochondrial branch chain alpha-keto acid dehydrogenase complex. Why alpha-keto acid? Because the alpha-keto acid is here, branch chain is there, that's why its name is branch chain alpha-keto acid, a dehydrogenase complex. This is a complex means there is some complexity in that, it is not that a single enzyme. So it is having a combination of 3 enzymes, that I will tell you, that name you have to remember and you have to memorize. So for this step I have told you that first step is transamination. For this one enzyme is there, single enzyme, that will convert all these 3 valine, leucine and isoleucine. All will be converted to their corresponding keto acid. Okay. See, what type of enzyme is a transaminase enzyme? You, just, I am asking you. Next. What type of reaction does it catalyze? Transamination. Can anybody tell what is the classification of enzymes? So these I am going to ask in the examination. In the Viva I will definitely ask. To each and every student giving a reaction. Because that is basic information you must have. Otherwise you will not understand what reaction it is catalyzing, what is the basic purpose. And I told you, if you don't remember, you try to, since your mobile is in your hand, you write down the name and try to find out the EC number. What type of reaction does it catalyze? Transamination. During transamination reaction I explained in detail, but still you did not bother to listen. Neither you opened your book. It is difficult to teach you. Suppose if I say something wrong, you will not be able to detect whether I have told something wrong to you. And now from next class onwards I will tell something wrong to you, and I want to have a reaction after 5 minutes whether somebody is raising this point, that I have told something wrong. It is something serious, very serious. I am really worried. Don't you think who have attended the transamination class I have explained? Some of you have answered. Even the liage, they have answered. At least one student I remember from the first bench told. Yes, he told, I think. You told liage. Hydrolyzed liage, somebody told. So this is transamination, it is transferring an amino acid. So it belongs to the second class. So EC number is 2. So you have not opened the book, you have not opened the chapter of enzyme at all. So please be very serious, otherwise you will fail, you are going to fail in the first internal assessment examination. No more simple question will be there except classification of enzymes. And what numbers, extra numbers will indicate, that also will be asked. Anyway, so transamination is there, that first reaction. You see, then this leucine will be converted into alpha-keto isocaproate. Remember, just note down one thing. I have written deliberately in the valine in the middle. So valine means isovalerate acid. Write down. So it will be easier to remember. Valine after transamination gets converted to alpha-keto isovalerate. And isoleucine, you remember that alpha-keto isovalerate is there, but only that in the middle there is a beta-methyl group is there. So, you remember this one, add only beta-methyl, then isoleucine will be there. And only one thing you remember for leucine, it is iso-alpha-keto isocaproic acid or isocaproate. As I told you, in the body, it doesn't remain in the acid form. That is a pH. So, usually it is present as pyruvic acid is not present, pyruvate, or lactic acid is not lactate, always you say in that form, acetoacetate. So, here we are saying that it is alpha keto isovalerate. But in the second step, remember, this will be leucine will be related to something somewhat like valerate. That is the reason I have highlighted it. Okay. So, that is a common branch chain amino acid transfer amino. See, you see here is a transfer of amino acid group of all three amino acids to alpha keto acid by a single vitamin B6 dependent branch chain amino acid transferase. But there is a specific amino acid for valine also. So, for valine there is a specific transaminase and a deficiency of which will cause a disease. That I will tell you at the end. So, as I told you, this one is a mitochondrial branch chain dehydrogenase complex, means it is not a simple thing. Now, you see the complexity. Now, you note down this one. There are 5 components of mitochondrial branch chain alpha keto acid dehydrogenase complex that you have to remember, memorize. So, there are 3 enzymes, one is E1, E2 and E3. One is decarboxylase, that is causing the decarboxylation reaction, and there is a trans-acylase enzyme, and there is a dehydrogenase reaction. So, this dehydrogenase basically is not a simple dehydrogenase, it is having an included decarboxylase and trans-acylase enzyme along with it. And there is another 2 enzymes, there are another 2 enzymes like it is something akin to your pyruvate dehydrogenase complex. You try to compare between the 2 after going back to hostel. There you open the pyruvate dehydrogenase complex enzyme and you will find a good amount of similarity between the 2. Maybe one question will be asked, tell me something, some analogy between pyruvate dehydrogenase complex and branch chain alpha keto acid dehydrogenase complex. And if you remember that thing, only thing is that some names are different, nothing more than that. So, that requires also TPP, that requires lipomide, that requires FAD. NAD is also there. PDH complex kinase and PDH complex phosphatase, these are allosteric enzyme. Basically, they are playing regulatory role. Kinase means phosphorylation. When the enzyme is phosphorylated, then means it is inactivated. When it is a dephosphorylated, so this is an activated. So, this needs to be regulated to allow the branch chain catabolism to occur. So, there must be some amount of regulation and this regulation is built in within the enzyme complex. So, when the enzyme will be active, when the enzyme will be inactive, means it will not allow the reaction to proceed further. Okay. Now, this is the reaction is, I told you transamination, then there is oxidative decarboxylation and the dehydrogenation, and finally these structures are produced. You need not remember these difficult names, these are not required as well, because disease you have to remember P's and Q's, everything. That is what I will tell you. So, only thing we wanted to highlight here, see decarboxylation, transamination is there. So, keto acid is formed. So, keto, this carbon dioxide, you see here, this decarboxylation, the carbon dioxide is out here from here. So, decarboxylation means this is forming an Acetyl-CoA, and then there is a dehydrogenation. Now, if you look at the leucine, it will ultimately broken down, note down the breakdown product of leucine is acetoacetate and acetyl-CoA. You make a dot, I mean some arrow, and write down leucine is finally broken down into acetoacetate and acetyl-CoA, and because of the acetoacetate it is a ketogenic amino acid. Valine, if you look at the valine, now valine is producing Succinyl-CoA at the end, and Isoleucine is producing Acetyl-CoA and Propionyl-CoA. From Propionyl-CoA, Succinyl-CoA can be formed also. So, that is the reason why it is a glucogenic amino acid. You write down the end product only. Write down here leucine, alpha-keto acid, dehydrogenation reaction, go straight down to acetoacetate and acetyl-CoA and Succinyl-CoA to acetoacetyl-CoA and propionyl-CoA. Bas, finish. Because there are difficult names are there, it is difficult to memorize and don't bother to remember also. Now, this is you note down each and every point. So, metabolic disorder associated with BCAA catabolism is Maple Syrup Urine Disease. This name you have to remember. Now, what is maple syrup? Maple syrup is not available in our country. It is a foreign, so whatever diseases you will see, strawberry-like appearance, this maple syrup, since these were invented or discovered by the foreign people, they try to name whatever things they have in their vicinity. If we had discovered something, we would have named something else. Now, out of sheer curiosity, I wanted to know what the maple syrup is, because there is no way how we can know this maple syrup. I heard this disease during the first year class also. But maple syrup means it's a smell of maple syrup, or it is like a burnt sugar. So, different types of maple syrups are available. It is something just like our date molasses or date jaggeries. It is produced in the same way. Rubber tree, if you remember how rubbers are collected, it is something like this, but date juice or palm juice is collected in a different way. So, what is done here, you see the hole is made in the tree and the juices are collected, and color of the juices is just like our date juice, and then it is processed, boiled, and finally something like molasses or this date molasses or date jaggery is prepared, and it is prepared in various concentration, and it is definitely sweet. A child is taking the sweet directly from the tree. That we also have done in the dates tree, because this type of juice were prepared at our house on the banks of our pond. The dates tree were there. So, the smell of the urine is because of the smell of the urine, it is called a maple syrup urine disease. So, there is an impaired function of that alpha-keto acid dehydrogenase complex. As I told you, alpha-keto acid dehydrogenase complex. So, dehydrogenase complex is here, decarboxylase, deacetylase and dehydrogenation. If there is a block, then this keto acid and the amino acids level should be high, and they will be excreted in the urine, and that urine will produce a typical smell which will be like maple syrup. That is the name why it is called a maple syrup urine disease. Okay. Now, this chart I told you earlier, again just for remembering. So, you see decarboxylase, transacylase and dehydrogenase. Now, you note down here. So, these three enzymes I have told you, it is there. Now, branch chain alpha-keto acid dehydrogenase complex, if there is an impaired function. So, since this is having three components, so three different types of disorders will be there. So, if there is a decarboxylase, the first enzyme, it is having two components, one is E1 alpha and E2 beta. E means enzyme. So, if the E1 alpha is deficient, then it will cause a type 1 type of maple syrup urine disease. If E1 beta of decarboxylase, then it is a type 1B MSUD. The second enzyme, if it is defective, then it produces type 3, type 2. And finally, the dehydrogenase which is there, that produces type 3. So, if you cannot remember anything, at least you write down, you just tell that alpha-keto dehydrogenase complex causes maple syrup urine disease. That will serve the purpose. But because of the three components, I have told you about the three different types of disease. Okay. So, there will be a mutation of the encoding gene. So, the enzymes will not be able to function normally. So, there will be a subnormal function, it can be total, it can be partial also. Okay. Have you written completely? Note down first, I will tell one more points and then I will close. This part is not available in your book. Maybe you open the Harper books it is there, and from Harper I have taken it. So now this transamination and oxidative decarboxylation, you see plasma and urine levels, as I told you repeatedly, 3 branch chain amino acids and their cognate alpha-keto acids will be increased. These amino acids and their cognate keto acids. So, these will be keto acids will be there. For leucine there will be separate keto acid, for isoleucine there will be separate keto acids. So, that is their cognate keto acids. So, they will be produced, okay. So, because block is here. So, the reaction cannot proceed further. So, signs and symptoms, what will happen? The excess amount of these amino acids will be there in the blood and it will interfere in protein synthesis, and brain will be worst affected because brain protein synthesis will not occur and there will be a variety of neurological symptoms. I mean, these neurological symptoms are common in many diseases, but whether it is specific for this maple syrup urine disease, it is very difficult to diagnose. The smell of the urine will point. So, first and foremost, there will be a complaint from the mother that, or if you examine the baby closely, if you try to smell the baby, which the smell of the newborn baby is very much varied. It varies from person to person, varies from races to races. And the smell is not that agreeable, newborn baby. So, maple syrup odor of the urine will be there, and since there is a high accumulation of ketone body, keto acids, so there will be ketoacidosis. So, acidosis will be there, and this acidosis will be due to keto acids. And neurological derangements, many things you can write down: seizures, convulsion, behavioral disorder, lethargy. I mean, a newborn baby cannot tell anything, so you will have to have an assessment that is some amount of problem in the neurological dysfunction, and mental retardation is very difficult to diagnose in the neuron period. The disease has to be diagnosed within the first week of life, otherwise considerable brain damage will occur, and the baby will be not only a burden for himself or herself, it will be a burden for the entire family. So, this disease has to be detected within the first week of life, and all inborn errors of metabolism are to be detected within the first week of life, and treatment is to be started to prevent complications. That you must remember. Those who are trying to plan to have a career in pediatrics, please read biochemistry very thoroughly, because you have to be very much vigilant after the baby is born, whether he is having inborn errors of metabolism. These diseases are very rare. Only thing is that if you know something, then only you will be able to detect, otherwise not. Otherwise not, it is not possible. So, early diagnosis will be urine levels of these enzymes will be very high as well as the blood levels, and that can be diagnosed by, nowadays it is available, it is in Tandem Mass Spectrometry. A number of tests like 40, 45 tests, even 100 tests can be done. You will get the report within an hour or so. Of course, it depends that the moment you send to the laboratory, it will not do it, because they will need some amount of sample collection, because there is a running cost of the machine. Because of that thing, the report you might get in the same day itself after a few hours, or maybe within 2 to 3 days, because for a single sample the machine will not be run. It is a very costly machine. I mean, ranging good machines cost somewhere around 2 crore rupees. At least more than 1 crore will be required to get a good quality machine and high output machine. An enzyme analysis, it is easier said, but it is difficult to do it. From where you will get the sample? So, treatment will be dietary restriction of these 3 amino acids. Now, these are essential amino acids. You cannot omit the entire amino acids, because protein synthesis will be hampered. So, there will be some amount of restriction, and it is a very difficult situation, because the baby is within first week of life, mother's milk is the staple food, and you have to restrict also this branch chain amino acid. It is really a challenging pediatric care issue, I must say. But at least for your examination purposes, you write down the dietary protein restriction, except protein mixture containing leucine, isoleucine and valine. Now, this is the maple syrup urine disease. You see, block is here, branch chain keto amino acids, all 3 amino acids will be blocked. So, the baby will be normal at birth. After some time, after 2 to 3 days, there will be feeding problem, there will be vomiting, because the disease has started appearing, there will be muscular rigidity or seizures. As I told you, these neurological problems present in diverse ways. So, first stage is basically feeding problem, you cannot feed properly, mother. There will be vomiting also. because brain is not functioning, and then muscular rigidity because neurological problem is there. The hands will not move the way it should, there will be some tightness, and then there will be seizures, means convulsion. If you look at the urine, you can do a test, then it will be positive. If diagnosed early and management can be instituted within the first week of life, at least good quality of life or near normal life can be given to this baby. Now, Tandem Mass Spectrometry was not there during our time, now it is available, but in a very limited center. Now, look at this level of this MSUD report. See, there is a reference range for all parameters. So, the advantage of Tandem Mass Spectrometry is that multiple analytes can be combined together. I mean, analyzed at a time. So, you see, look at the level of alanine here, the normal level of is 74 to 603, the level is here high, that means there must be, it is not, it is 74 to 603, level is slightly low. But if you look at the arginine, it is 9.94, the figure is up to 72. But look at the level of leucine, isoleucine, the normal level is up to 100, and in case of MSUD, it is 1574. So, diagnosis is very obvious. In Valine also, you see the normal range, it is given like this, but the value is on the higher side. So, this is a case of typical case of, diagnosis is almost confirmed because you have measured the concerned analyte. Now, once you start the treatment and you measure the amino acid level, it will be decreased. That is how you will monitor the patient. So, without Tandem Mass Spectrometry, diagnosis is difficult. But earlier it was where this Tandem Mass Spectrometry was not there when this disease was diagnosed, actually invented. So, they have done it in some other older methods. So, there are these you write down. These 4 types of other disorder is there. There is intermittent branch chain aciduria. It is due to some or partial loss of alpha-keto decarboxylase component. If you cannot remember alpha-keto decarboxylase, write down Alpha-Keto Dehydrogenase Complex. But specifically it is due to alpha-keto decarboxylase. There is a partial loss. So, some functions will be there. That is the reason why intermittent branch chain ketonuria will be there. When the person will take excess amount of protein, then only it will happen, and the child will grow normally. There will be no problem of the growth and development because partial loss is there. Isovaleric acidemia, this I told you it is related to leucine. So, isovaleric enzyme dehydrogenase, here it is a decarboxylase, here it is dehydrogenase, remember. So, normally nothing will happen, the person will also not know that he is having this disease, but when an excess amount of protein is ingested, then there will be vomiting, acidosis and even coma because of the accumulation of isovalerate. And this isovalerate will produce a typical odor, that is a cheesy odor, but it does not occur normally. When a protein food will be taken in excess, then only it will happen, because there is some problem with the specific enzyme. Okay? And last one is hypervalinemia. Here valine is there. I told you alpha-keto, there is a common transaminase enzyme, but if there is a specific enzyme is for valine, so valine transaminase, if it is absent, then it will cause hypervalinemia. Okay, it is a deficiency of valine transaminase, but since these two are paired, so their level will be normal. Is it clear? Is this point clear? Alpha-Keto Acid Dehydrogenase amynase, this is a common enzyme. Branch chain transaminase is a common single enzyme disorder. This will cause transamination. But if there is a specific enzyme which is blocked, then only for valine, then it is called hypervalinemia. So, these at least MSUD, if disease is there, try to write down the entire thing, whatever is possible, at least first part you must write down. So, tandem mass spectrometry, I have shown it to you. Newborn screening, much is talked about, how a newborn baby will be screened. So, every state, every nation has a guideline, but unfortunately in our country there is no actual statistics and it is not done in all the places. Some tests which you used to do in your undergraduate, even during your postgraduates, were the mainstay of diagnosing the cases, but at a late stage. So, only thing is that the 4 points needs to be satisfied before a newborn screening is made. I mean, as a health policy, that is an acceptable treatment protocol that must be there in place, and that the change outcome for patients diagnosed early with the disease. One classical example I am giving you, congenital hypothyroidism. If in a case of congenital hypothyroidism, if thyroxine is given in the very early stage, permanent brain damage can be avoided and the child will grow normally. So, that is an accepted form of treatment. That accepted form of treatment there, and understanding of the condition's natural history. As you will read further, you will understand most of the diseases are cured normally. So, that is what is called natural history. Some diseases will lead to deterioration, finally lead to death. Some diseases will have an intermittent period of fluctuation. So, that is what is called natural history of the disease. So, that natural history of the disease must be well known. Third point, an understanding about who will be treated as a patient. But because for that you have to make a confirmed diagnosis. Otherwise, you cannot treat arbitrarily. Fourth point is that the screening test must be reliable. Screening test is not a confirmatory test, but it must be able to confirm the disease. An affected person will show the disease, but unaffected person will be negative for that. That point you must remember. And challenging task is IM blood collection and samples. That is another issue. So, normally blood gas can be done, blood glucose can be analyzed, blood ammonia and urine ketone body. These are newborn screening. So, at the earliest possible time, this disease has to be diagnosed, preferably within the first week of life. Day before yesterday, I talked to a senior pediatrician. I told you 3 years senior to me. I told you what is the status of newborn screening in India. What he told, the summary is that for congenital hypothyroidism it is done, congenital adrenal hyperplasia, galactosemia and PKU, but at different centers. It is not done even in all centers. So, babies are born everywhere. The screening tests are done only for these 4 diseases and there are different centers. So, common diseases identified in India, there are many, but the tests are not done everywhere, even in medical colleges. So that is a big problem. Knowing is not sufficient. Implementing it so that you make a meaningful difference in the patient care. Only discussing in the theory class, answering an MCQ question, getting a good mark is not going to help the patient in any way. So, Tandem Mass Spectrometry has really revolutionized the newborn screening method, but it is costly and available in a limited center. And next generation sequencing is a high output DNA mutation diagnostic tool. I wanted to have a training at Coimbatore because of the Indigo fiasco. I could not attend this training because a large set of genes can be analyzed at a time and within a short period of time. And now if you combine Tandem Mass Spectrometry and next generation sequencing together, at least not only these known diseases can be diagnosed, but many unknown diseases will be diagnosed, not yet discovered, not yet known. So we will have a really good revolution, and as a biochemist, instead of doing this type of job, we are supposed to be involved in this type of work. But Tandem Mass Spectrometry, next generation sequencing, patient analysis, and then further step of medical care, that will be a real clinical oriented biochemical approach to management. So, while you will be, while you will pass, I think by this time the combination of all these things will be available and you will be able to diagnose that disease very early or maybe within the first week itself. So, I told you during your urea cycle class this step, so those who have missed it, for them I am telling you the standard method for Tandem Mass Spectrometry and NGS testing is a dried blood spot specimen. Usually those companies supply this type of strips where from the heel prick drops of blood are collected and the diameter is usually about 8 to 14 millimeter, from where 3.2 millimeter punches are taken and they are eluted, samples are collected and then Tandem Mass Spectrometry is done or DNA analysis can be done from that in the NGS platform. So that a huge number of, a battery of genetic tests can be diagnosed. So, either Tandem Mass Spectrometry that will be less costly, NGS will be another costly affair. Now, there are many affluent person who can have a combination of both the things, so that at least nowadays we are having only 1 or 2 child and there is a very spacious child. So, we do not want to lose any child. So, this can be done. So, earlier there is a number of tests that were in place and this will be basically lost into oblivion because of the advent of these 2 tests. So, what I tried to do in 1993, I have made an humble attempt to diagnose G6PD deficiency for diagnostic purposes for the patients at Durgapur Steel Plant Hospital. At least some patients so that I can diagnose these cases. A problem is that if you start anything in a government hospital, there will be demand that why I will not be tested. So, a huge sample load started coming to me. It is a very low cost investigation and I wanted to extend the effort to persons who will be really requiring particularly anemia cases, undiagnosed cases. But then they started sending the samples for almost all babies and it is very difficult for me to do an additional amount of test which will require 6 ml of blood and do the test and report it. So, at one point of time I had to stop it. So, my craze for this thing did not go. Since this is a neurological problems are very much predominant in the newborn screening cases, I will take 2 or 3 more minutes, maximum 5 minutes. to tell you the state of affairs. NIMHANS, that is National Institute of Mental Disease and Health and Neuroscience, this is the National Apex Institute in India. So since neurological problems are there, then I thought that I must get trained myself for detection of inborn errors of metabolism, and deliberately I opted for the third batch, and I went for 10 days training, and to my utter surprise, I am sorry to say that even in the third national workshop, the tests were not standardized, and I wasted 10 days time at NIMHANS without getting anything from them. So even in the highest center, that was the state of affairs in 2004. So don't think that everything is, fine, that a big institute I have went there. The tests were not standardized. The samples were not there. So during urea class, I told you, I am just repeating you because there are many students who are not there. So when the baby is born, then the problem is there. It's a difficult situation. We have to prevent the birth of the affected baby for genetic diseases. So that's why I made an humble effort in the year. I conceptualized in the late 90s how to prevent it. So I wanted to detect it before the baby is delivered and while it is in the womb. So I made an ambitious project for Eastern India. The center will be at Durgapur Steel Plant Hospital under my principal investigatorship, and I got the project approved from Lucknow. Someone of you are from Lucknow, I know. It is a very difficult task for me to get it approved from the SGPJ, and then I submitted to Ministry of Department of Biotechnology, Ministry of Science and Technology. Because even in today, in 2025, it is not in sight. I wanted to do this study center in 2002 so that at least unborn child who will not be delivered, the mother will have an option to get the baby aborted. But that is what is prenatal diagnostic detection. For an unborn child, I don't want to create the problem after the baby is born. But unfortunately, this project was submitted only 21.5 lakhs rupees. I wanted to start the setup. But because of the rate tapism, I had to get a clearance from Steel Authority of India Limited. It was a very difficult situation because I am going to extend the offer to general people. It was published in Hindustan Times, displayed in SGPJ that the SGPJ is going to set up its center at Durgapur, but at the end, project was not sanctioned because of the rate tapism. I did not include HOD in my as an associate this investigator name you see, the HOD name is not there because he is an anthropologist. I did not want her. So that is the reason she created that problem and got the project cancelled. So finally I published this paper. You can search it in the internet. I did not want, see, if you want to get the baby sample, either you have to do a chorionic villus sampling. Now for chorionic villus sampling, there is a problem that if you do it in the early stage, you don't know from which part of that particular zygote which organ is developed, and there is a high chance of babies the abortion of that mother, and there will be malformation, and we need a gynecologist, we need an obstetrician, ultrasonologist. To overcome this, I wanted to do it from the peripheral blood of the mother, and that paper is there. It is still on the paper, it is not in real force today. So if somebody is interested to do something, systems are there in force not to allow him to do that thing. If you don't do, he will be criticized, you are not doing anything. If you want to do, that job will not be allowed, and this is a glaring example. With this thing, I conclude. I think I have talked too much, but I am telling you, try to do something. At least if you attempt for 10 jobs, you will be successful in at least for one job. think something beyond what is written in the textbook, think something well ahead of time. I thought 25 years back about this project which is not in sight today. Even the usually gynecology, I inquired, sir, do you have a system? No. Pediatrics department, you see the state of affairs, newborn screening, only 4 diseases, that is on the limited centers, but that center would have catered the eastern India, and my blood was really full of energy during that point of time. I could have developed the center, and this could have been one of the best center in India. Only 3 places are there, these tests were done: Lucknow, Delhi and Hyderabad.